and method for effective early identification and prevention of ToBRFV. Very challenging title.
So thank you very much. Can I use the microphone?
Yes.
And to move between slides.
You have just to use the
Okay.
So you can sit or stand.
Okay, I will try to stand.
Okay.
So can you hear me? So good afternoon. I'm Natasha Mehle from National Institute of Biology from Slovenia. However, today I will present also the work that was done at FERRE at UK and Santa in Belgium. So my talk, as it is from the title, I will talk about the possibility how to early detect tomato brown rugosa fruit virus, because this is actually important to prevent further outbreaks. So, as you all know, tomato brown rugosa fruit virus is very stable virus that can spread very efficiently and cause serious diseases on tomato plants worldwide. It can be transmitted through the seedlings and by mechanical contact. And we also confirmed that water could be also one of the possible transmission pathway. In Slovenia, we detect the RNA of this virus in several different environmental waters, such as river, ponds, wastewater, and also in waters used for irrigation. And what is the most interesting is that we detect the RNA of this virus in a wastewater sample that was taken in 2017, so much before we found first infected plants with this virus in Slovenia, which was in 2021. The source of water contamination with this virus could be various. And one of the possible sources we also experimentally confirmed. This is that tomato brown rugosa fruit virus infected plants can release infectious virus particles into water through the roots. We also confirmed that tomato brown rugosa fruit virus can survive in water stored at room temperature for up to 4 weeks, which of course depends on the virus concentration in the water as well as on the environmental conditions. And in this slide, I also need to mention that we were able to detect the RNA of this virus with real-time PCR much longer. Here are the results for 9 weeks when the water was stored at room temperature, but similar CQ values we obtained also when we tested the water after 15 weeks. We also experimentally confirmed that water can be used also for the transmission of the viruses, so to infect the new plants. In example, when we imitate the hydroponic growth system, we confirmed that tomato brown rugosa fruit virus from the nutrient solution can infect healthy tomato plants through the roots and eventually spread to the upper parts of the plants, where it can be detected after 1 to 6 months, which is dependent on the virus concentration in the water as well on the severity of the root damage. Similar results we also obtained when we irrigate with infested water the plants grown in the substrate. So from these results, I would say that water is very important potential risk factor for spreading of tomato brown rugosa fruit virus. Therefore, I would highly recommend to test water if it is used for irrigation to prevent new tomato brown rugosa fruit virus outbreaks. In addition, water monitoring could also serve as a tool for early virus detection. And this last statement I will now explain on tomato commercial greenhouses from different countries in Northwest Europe. These are actually the results from Santa Terrare from Belgium, where they did routine monitoring of drain water. Here are the results of 4 greenhouses. In greenhouse A, B, and C, there was no previous tomato brown rugosa fruit virus outbreak, while in the greenhouse D, there was 1 year before they did monitoring tomato brown rugosa fruit virus outbreak. So and they took not just drain water, but also leaf samples and tested them by real-time PCR. So in example of greenhouse A, they tested leaf samples from 8 rows. And in week 5, suddenly they got positive 1 leaf sample with tomato brown rugosa fruit virus. However, first symptoms they were able to observe only a week after, so in week 6. But what is interesting is that they were able to get decreased signal with real-time PCR when they test drain water, also a week before they were able to see the first symptoms. Similar results they got also in the greenhouse B and C, where they also observed this decreased CQ value for tomato brown rugosa fruit virus in the drain water a week or 2 before they observed the first symptoms. However, in practice, it is possible that with real-time PCR in the water, we detect just the residuals of tomato brown rugosa fruit virus. And this is illustrated in the case of greenhouse D, where there was 1 year before the outbreak, and the year of when they did the monitoring, there was no new infected plants. However, with real-time PCR, they got a CQ values constantly, but these CQ values during the time increase. So lower concentration of the viral RNA. According to our experiences, tomato brown rugosa fruit virus may be present in water samples at very low concentration. Therefore, for reliable detection, we need diagnostic methods. These are usually real-time PCR combined with one efficient concentration technique. For routine monitoring of water, different environmental water samples for different viruses, we use monolithic chromatography to concentrate the viruses, and then different techniques for detect and to identify the viruses. In this procedure, we can analyze up to 5 liters of water samples. The first step is filtering, so to remove large particles in the water. Then the next step is concentration step. We use monolith column from BS separation. And after concentration step, we extract the RNA with KIA-AMP viral RNA mini kit from QIAGEN. So this procedure, we found that efficiently concentrate also tomato brown rugosa fruit virus from different environmental samples. For example, in a water sample from the last year, after concentration step, we get the signal with real-time PCR for tomato brown rugosa fruit virus with CQ28, while before concentration step, there was no signal with the real-time PCR. Using this procedure, we can also get enough RNA of the viruses that we can do then further confirmation either with HTS or Sanger sequencing. So this monolithic chromatography, it is very efficient for the concentration of different viruses, including tomato brown rugosa fruit virus. But it requires complex equipment that is not available in many laboratories. Because of that, we, in collaboration with FERA from the UK, evaluate other possibility for the concentration. And this was ultrafiltration with Centricon plus 70 centrifugal filter units, short-lived centricons, which can be applied in any laboratory with bench-top centrifuge. This method, we found that also efficiently concentrate tomato brown rugosa fruit virus, as you can see from the table with the CQ values after concentration, much lower CQ values compared to the samples where we did not any concentration step. However, main disadvantages of this method is that we can concentrate only up to 100 milliliter of water sample. So comparing with monoliths where we can concentrate up to 5 liters. So I would recommend this concentration step, for example, for water samples, where you can expect higher concentration of the viruses, like water that is recirculating in the hydroponic system or wastewater. Then, sending of the samples to the laboratory takes time. And in addition, it's not easy to handle bottles with water, and it's very hard to send them by email. So our aim is that at least this concentration step can be done on site. Theoretically, this is possible with centricones or with sea monoliths, as you can see from the picture, but then we need to bring all of those equipment in the field, which is not very easy to be done. So now we are working on searching other alternative methods for the concentration. And we found that ultrafiltration units from InnovaPrep, like large volume concentration kit and concentrating pipette, are very promising to be used on site. In addition, with these procedures, we can concentrate larger water volumes. We did not test these two procedures yet for tomato brown rugosa fruit virus, but we found, for example, that it works very well for a similar virus, pepper mild mottle virus. So I think it should work for tomato brown rugosa fruit virus as well. So why we are working with that? Because we believe that implementing early detection strategies together with regular water testing and biosecurity protocols enhance the ability to mitigate risk, reduce economic losses, and prevent further spread of tomato brown rugosa fruit virus. Strengthening water-based surveillance as part of integrated management approach is essential for controlling this persistent viral threat. So just at the end, that you will not think that we are just working with the methods how to detect the virus in the water. I would just like to mention that we are working also on solutions how to inactivate the viruses in the water. So we are working with hydrodynamic cavitation with cold atmospheric plasma, which we found that are very efficient method for inactivation of different viruses, also viruses that are similar to tomato brown rugosa fruit virus, like pepper mild mottle virus, and also for potato virus I, for example. We did not test yet this for tomato brown rugosa fruit virus, but are very promising methods. So the results that I presented today have been done in the frame of the National Research Applied Project, which is financed by the Slovenian Research Agency, company BIA, and by the Slovenian Ministry of Agriculture, Forestry and Food. So at this point, I would like to thanks to all of my colleagues that are listed here to work on this project, and many thanks also to colleagues from FERA for really nice collaboration in this project, and many thanks also to colleagues from Centre Terrare from Belgium that they allow me to present their results as well. And of course, thanks for organizer to invite me, and thank you for listening.
The Israeli point of view, because everyone can tell the story of ToBRFV from different point of view. This is the damage of ToBRFV. And let's start. So, Tobamovirus species number 37. So you can see here the images. What is nice, people that working with microscopy, with STEM, you can see the size of the particle. They are not 300 nanometers like the other tobamoviruses. So different sizes of particles. So this is quite unique for ToBRFV. And the story started in 2014. At this time, we didn't know that it's a ToBRFV that will change completely the tomato industry worldwide. So here we saw the typical symptom. At this time, we didn't know what it is, what it looked like a tobamovirus. The symptom, like a TMV or TOMV, tomato mosaic or tobacco mosaic. We've been surprised. At the beginning, also, we found with mixed infection with tomato spotted wilt, the TOSPO virus, and the typical symptom with the yellowing spot on the fruit. And this is the beginning of ToBRFV in Israel. After that, so now, this is quite update from the EPO, the global database. We can see that it's distributed worldwide. So in Europe, also Central America, South America, and also recently in Australia. So it's a big deal in tomato industry. And also the PRA, the old PRAs that I understand that now in Europe, it's not a quarantine pest anymore, if I understand correctly, that it changed, the rules changed. We tried to put pieces in the puzzle of ToBRFV spread, so we divided to primary inoculums, that it can be seed, soil, and weeds. We can think if water is part of the primary inoculum. And after that, there is the secondary spread. So it means any kind of mechanical handling during the handling of the plant, the trellising and the pruning. Also, pollinator can contribute a little bit for the spread, but inside the structure, inside the greenhouse. There are a lot of questions that are still under research. And today I will speak only on this area of the rectangle in red, so only on the soil part. And I think in VIRTIGATION, we put a big effort to understand how the virus preserve in the soil, what we can do to manage the virus preservation in soil, and some technique that can be for management. So here you can see the beginning, reducing the primary inoculum in the soil. So after several years of monitoring the number of the primary source, what does mean primary source? It means that we go to commercial grower and we count after 2 or 3 weeks the number of infected plant. Before the trellising, before touching the plant, and we understand that it's about 1 to 3%, but it depends the type of the soil. So this kind of agriculture that in Israel we grow mostly in the soil, directly in the soil. I know that it's been surprised for people in Europe that grow in substrate and hydroponic, but we grow mostly in the soil. And soil, after the previous infected cycle, is contaminated with the virus. And it means that the second cycle of growing will be infected. And the number, it's between 1 to 3%, depend the type of the soil, because sandy soil wound less the roots compared to heavy soil. So the phytosanitation is really, really important. We can purify the virus from the soil. We can see the virus particle in the soil. And you can see some practice that you use the polyethylene film to cover and to minimize the contact with the new seedling touching the soil, the contaminated soil. So this is common practice that used to minimize the infection at the planting time. So what you can see here, it's after 2 to 3 weeks post-planting, we can see some typical ToBRFV symptoms. So this is the time of infection through the soil. And you see the big difference if you get infection through the water, that it takes 6 weeks. So here it's 2 to 3 weeks. Of course, it depends the time and the temperature. In the summer, everything runs faster. In the winter, it takes 1 week more. Typical symptom. And before the trellising, you can see the trellising rod, but still without touching the plant. So it means this is a primary infection source of the virus. So at the beginning, we built sort of experiment. We use the R&D centers that we have for run a big experiment in R&D center in the south. So we wanted to check some chemicals that can be used for soil disinfection. We use stabilized chlorine, different brand of stabilized chlorine to disinfect the soil. So in this kind of set of experiment, we inoculate the soil, you can see on the left side. And after that, we pour the inoculum into the planting pit. So like that, we can be sure that we contaminate well the planting pit. And also, we truncated the roots to increase the wounding. And after that, we have a nice system to inoculate the plant, because the point in this experiment is to keep a good positive control. And if the positive control is working, now we can check the different brand of chemicals that can be used. So here is a summary of 5 different experiments, different time period. So we can see that the control is something like 38% in average. So it's good. It means we can compare to different brand of chlorine and chlorine TSP. But still, it's not abolish the infection, but it's minimize it quite nicely. If we compare all the 5 experiments with the control, we understand that it's something. And I can tell you that in Israel, we use it, and the grower use it because they use the chemical. They apply the chemical 1 day before the planting through the irrigation system, and it's quite cheap treatment. After that, we try to also learn about the infection through the soil, and we build sort of experimental system. Understands that the time that the infected plant is growing and the root grown inside the soil, so it affects the number of infected plant. So if we grow the inoculum for 30 days or 90 days, we can see, let's see if the pointer, go back. So we can see that the average of infection after 30 days that we grow infected plant can be 3%. And if we grow for 90 days the infected plant, and after that uproot the plant and plant a new one, especially when we truncate the roots, we can get even higher infection. So this is our system, check different chemicals that can be used. Also, we try to learn the longevity of the virus in the soil, and we collect the soil from infected plants. So we uproot the plants and we collect all the soil and we build this kind of a pile, and we have a wet pile and a dry pile. And every time period, we took a sample of the soil for 50 plants, truncated the root and plant the 50 plants. And like that, we can calculate the time that the virus decreased in the soil. And we've been surprised that it's quite long. After we can say 100 days, you can see the wet pile in the middle, that after 7 days and 100 days, we can see decrease. And each time point, it's 50 plants that been checked. And we almost got the zero, but then we repeat again time period and we saw it again. So we understand that the virus can survive infection in the soil, something like 200 days, to be on the safe side. So now we don't have any recommendation for the grower, because if a tomato grower wants to grow tomato, I cannot tell him leave the greenhouse for 200 days. It's not practical. So we move on to something that can be useful. And we try to learn the mechanism of the disinfectant, and we choose the chlorinate TSP for the on. And in this set of experiments, we try to see the effect of the pH on particle morphology, on genome integrity, and at the end on the infectivity. So you can see the range of the pH. You can see that at pH 1, the image on the right side, the left one, you can see that the particles look okay. And this has been surprising, because in the literature, everyone says that low pH can affect the particle morphology, but we checked it several times and we saw the particle look quite okay, and they can be infectious. So we go in the pH to pH 7, the particle morphology look perfect, but we've been surprised what's happened after pH 10. So you can see here that in pH 11, we can see the breakdown of the particle morphology. We can see some disk, but there is no particle anymore. So the image on the right side below summarizes all the experiments that we did to analyze and characterize what's happening in different pH range. So we can see the 10 effect, the 10 visualization of different pH. We understand that at pH 11, there is no more particle. On the left side, we use the PCR to amplify large segments, so it's in most of the genome, something like 6,000 base pairs for amplification, with some control for a smaller amplification. We understand that above pH 11, we break down the particle, the genome integrity is affected. Interesting to see what's happened to the cold protein. So you can see in the chromacy, after pH 11, we still can see the cold protein subunit, but at 12, we see breakdown of the cold protein subunit. And this is a complete inactivation of the virus. So in order to inactivate the virus, first we need to break down the particle, after that to break down the genome, and basically we don't need to break down the cold protein. No need, but this is what occurred above pH 11. All of that we did with a bioassay. It's meant to inoculate a tobacco plant, Nicotiana glutinosa, with the local lesion assay. We tried to apply our understanding for the chlorinate TSP. So it's a nice combination of 3% chlorine and 97% trisodium phosphate that is well known for soil disinfection. The trisodium phosphate is quite commonly used for seeds companies. So a combination of the two chemicals give a sort of synergistic effect. So because we can use lower presentation of the chlorinated TSP. TSP is commonly used in 10%. So now we can use 1 up to 3% for virus disinfection. We apply that in a soil experiment with a control to tap water, and we try also to increase the pH using the KOH, but basically we don't need to higher pH. So the chlorinated TSP alone give the pH for virus disinfection. So basically, we have 3 modes of inactivation. So we have the chlorine, we have the trisodium phosphate, and we have the high pH. So combination of that is work quite nicely for virus disinfection, and it starts to be used now in agriculture because also this kind of chemical is quite cheap. But we try to move not to grow directly on the soil. So it's the beginning of changing. The problem that here we use the coconut coir is that the grower have finished the 1 cycle of grow, and after that all the plants are infected. And basically you want to save the replacement of the coconut coir, and you want to continue for another cycle. So here what he tried to do, so this is what the grower tried to do. We tried to follow him and learn, and also to assist him with understanding what's going on with. So here it's a solarization, but we call it's not a true solarization because it's not open to sky. It's solarization inside the greenhouse. The polyethylene cover is still on the top, so it's not a perfect solarization, but still. So we cover the substrate with a plastic film. Maybe it's difficult to see. We collect some sample for 0 time to see that there is a source of infection. So here you can see on the right side the bioassay Nicotiana glutinosa. So the virus is inside. We connect a data logger to record the temperature, and you can see the line on the top. So it's mean the weeks the temperature inside the substrate was above 50. So it's okay. This is what he can get. And for him, so different data loggers. And we return after the planting, 35, 40 days, collect 100 randomly sample because we didn't saw any primary infection. So for the grower, it was perfect. And he continued to do that for 4 cycles. So this was in 2023, before everything started in Israel. But even now, he started the 4th cycle, and he's quite happy with that. It's quite cheap. So during the solarization, also the grower does this kind of metam sodium. It's for fungus that is the substrate. It's commonly soil disinfection treatment. So it's mean he combine the treatment. They do the solarization together with the metam sodium. This is quite nice, but now in vertigation, we try to do something more organized to control all the parameters. So solarization model experiment. So it starts with number 1, inoculation of plant, grow them for 3 months. After that, uproot the plant, collected the substrate, the grow base, also cover with all of them, and start different treatment, solarization alone and solarization with chlorinated TSP. Temperature reach above 50, sometimes 60 degrees, because we did the proper solarization. If you can see that they are outside. We can see the humidity inside. So it means the solarization came with irrigation on daily basis. So the irrigation temperature can reach 60, sometimes a bit more. So nice temperature. Maybe not enough for a small time period for inactivation, but in the long run, after several weeks, there is effect. Here you can see the experiment. also outside. And on the left side, you can see the experiment, but on the right side, you can see the bioassays that we try to do. So at each time point, we take back the grow bags, place them in the greenhouse, plant new tomato seedling, truncated the roots to increase susceptibility, and wait for 30, 40 days to see infection. So this is the bioassay. In parallel, we did bioassay on glutinosa, but also a real bioassay in tomato plant. But this is the result, the recent result from the recent summer. We are not happy with them. I can say the truth, we don't understand, but there is a trend. So we say, also the students that did the work say, I'm embarrassed to show the results, but we can learn something. So we can learn first about the bioassay, what we did in the tomato, because if I go back, you can see this one, it's too dense for a bioassay in tomato for ToBRFV. So for sure there is a contamination. On the right side, you can see all the plant, different treatment. This is one of the problems that can be explained why we are not happy with the results. But we understand that after 35 days, so the blue are the healthy plant and the red are infected, different time period, different treatment. We understand that the chlorinated scheme maybe is not working well with the solarization. This can be alone, no need to do them together. We understand that the plastic covering is very important. And we understand that after 35 days, there is some trend of something is working, but basically, it's not in accordance to the results that we saw for 2 years with the grower. So the experiments that we ran in the lab was not so perfectly, but in terms of to do that in practice in large scale, solarization is good. After 35, 50 or 70 days, it's perfect. In terms of massive agriculture, for science, we need to go in details. And what it's mean how to purify the sample from the coconut coir. So we try several methods for extraction. So the classical was to use the ultracentrifuge to get large volume and to precipitate, but this is something that we can do 6 samples a day or 12 samples a day. It's not practice. We have hundreds of samples to check the grow bag, so it's not good. We use evaporation in wood, chemical wood, so leave them open. It's evaporate quite fast. This is cheap, and we need that. We still improve that. Now we use also the sort of immunoprecipitation. We use the antibodies and the secondary antibody with the metal and magnetic beads to concentrate. But this is more complicated, not practiced in large scale. So basically to extract, leave it open to evaporate, and then to run the PCR bioassay. This seems like sort of a reasonable procedure for several hundreds of samples. So here the summary. Phytosanitation, if you go directly, if for growers that grow directly on the soil, is really, really important to remove all plant debris and also root material. Using sort of stabilized chlorine or chlorinated SP can be good. Solarization for hot countries like Spain and some Middle East countries can work quite nicely for 60, 70 days. It costs not so much. So for countries that have the sun, it's perfect. In terms of the science, we need to go into the details to understand how everything is working. So first to thank the VIRTIGATION project for the support. It was tough 2 years in Israel to do the solarization experiment because all the tomato industry is in the south. So it was quite challenging.
How difficult it is to mitigate the coronavirus infection, because this is a virus that is transmitted via mechanical means. So that means that it's very challenging to control the spread of the virus. So to avoid to have the virus in our crops, it's critical to avoid the introduction of the virus. So for this purpose, we must implement hygiene measures as eliminate the infected plants, wash the hands, and also disinfect all the tools that we are using with the crops. And also we must use certified virus-free seeds, and also if they are available, some resistant varieties. We can also implement subcultural activities like crop rotation, and also the soil disinfection methods, like the one that I'm going to talk about now. It's also a good practice to monitor the plants, looking for virus symptoms, and also to analyze the plant material in order to detect as early as possible the presence of the virus. And also we can use some chemical treatments, but today only some products are really effective against this virus. So Abim has already talked about the solarization, so I'm not going to talk a lot about that. Just to say that the solarization is a disinfection method that is chemical-free, and I use the solar radiation to heat the substrate of the soil in order to increase the temperature, and also to eliminate the soil-borne pathogens. And this is a method that is extended for some weeks, typically from 4 to 6 weeks during the hottest months. So this is a method that is used in warm areas like the Mediterranean area. This is a chemical-free and very environmental friendly and also very safe. So it's a really recommended method to eliminate the virus. To mention a disadvantage that it requires a high solar radiation, so it must be suitable for all the areas in the world. So the steaming process is quite similar, but in this case, the heat coming from the steam. So the temperature that is able to reach with this method is higher, from 60 to 100 degrees, but this method is short time. It's able to eliminate a broad spectrum of pests, but a disadvantage is high energy consumption, is more difficult to apply, and also is more expensive than solarization. So with this work, the main objective was to evaluate the efficacy of these disinfection methods, the solarization and the steaming methods, to eradicate or to mitigate the coronavirus from the substrate. So first, I'm going to present the results from the solarization method, the solarization trial that were performed in technology. So this trial was performed in the city of Almeria, that benefits from the city, that benefits from the Mediterranean climate, so it's very well suited for this method. So specifically, this trial was conducted in a multi-tunnel type greenhouse that is placed at the facility of the Experimental Center Technology. So to inoculate the plants, we selected one-to-one virus, the tobacco mosaic virus, because ToBRFV was a quarantine pest by the time that we conducted the trial. So that is the reason why we moved to the tobacco mosaic virus. This is a virus that infects tobacco, but also tomato, and that produces symptoms like the mottle and also the mosaic on the leaf. So for the inoculation of the plants, we prepared 2 different inoculums, 2 different solutions. One of them were prepared using the semi-purified virus, and the other one incorporated fresh infected leaf material. So we performed 2 different inoculations to ensure the success of the infection. The one on 15 days on seedlings, and the second one on 14 days on seedlings, by gently rubbing a cotton swab onto leaves that were previously dusted with carborundum powder. So on plant material, we used 2 different tomato varieties. One of them a susceptible variety, that is the moneymaker, and the second a commercial resistant one. And we also include in the trial the tobacco, as it's a susceptible host of the virus. So this experimental consists of 3 stages. The first one is a pressurization trial that we perform in order to obtain the contaminated substrates. A second one that is the solarization process. And the third is the post-solarization assay in order to verify whether the viral particles are active or inactive after the solarization process. So this is now starting with the pressurization trial. This is experimental design. So for this pressurization trial, all the tomato and tobacco plants were inoculated and then transplanted into the cocoa peat bags. And then all these bags were placed in the greenhouse as it's represented in this figure. So this is all the treatment that we have. We have an active control, that is the resistant tomato plants. We have 2 positive controls that are the susceptible tomato variety and the tobacco plants inoculated with the TMV virus. And we have also 4 different treatments that consist of the susceptible tomato plants inoculated with the T1 inoculum and the T2 inoculum. And this is an image of the tomato and the tobacco plants during the pressurization trial. So during the solarization project, this is the protocol that we follow. So this method lasts from September to November, 60 days. And the first step was cut or remove the whole plants. Here we wanted to evaluate 2 different conditions. We want to check or to evaluate if including infected material plants have an effect on the solarization process. So we split all the bags in 2, and in one group we included the infected material plant, that is the condition B, and the other one we ripped the plants and we removed all the roots and all the leaves. So next we introduce in all the bags the halogen to register all the temperature along the trial. And also next we cover all the bags with a plastic material, fill all them with water, and finally we close the aeration system and the windows of the greenhouse in order to increase the temperature. And then the post-solarization assay, the last stage of the trial. For this, we use new healthy tomato plants that we transplant on the cocoa peats, solarized cocoa peat bags. We observe the symptoms, if this appears some symptoms associated to the antivirus. And we also use some tobacco plants to inoculate them with the tomato plants that I have already mentioned, and we observe if there is some symptoms of the tobacco plants. So this is the experimental design. So for the post-solarization trial, we collected all the bags and we placed them in the greenhouse in 3 different areas. So in one area, we placed all the solarized bags without infected material. In another area, the solarized bags with infected, that includes infected material. And in a third one, we use new tobacco plants for the bioassays to inoculate them with the tomato leaves coming from the other areas. And also here, we include some new treatments that are healthy tomato plants, resistant and susceptible variety, and tobacco plants in order to check the normal growth of the plants. And to test the presence of the virus along the trial, we use the immunoreactive strips, and also at the end of the trial, we perform a PCR test in collaboration with Center in order to try to detect the presence in leaves and also in the substrate. So the results that we got with the personalization trial, we observed that tobacco and susceptible tobacco plants show severe and mild symptoms respectively after viral inoculation. So the viral inoculation was successful. And we also confirmed that all the plants are positive for the virus, and that means that the both methodologies of the both solution were succeed. So we were able to inoculate all plants using the both solutions. So during the solarization trial, we monitor all we registered the temperature inside the bags, and we observed that all the bags we reached a temperature above 40 degrees. And here we observe that those bags that include infected material plants, that is the one that is 24 days, were able to reach higher temperatures for more time than those bags that didn't include the infected material. So as I have said, we conclude that the fact that include the infected material help to increase the temperature inside the bags. And this is a very good point for increase the success of the disease infection process. So the result of the post-solarization trial, after solarization, all the new tomato plants grew up symptomless in these solarizated bags, but at the end of the trial, we observed that some of them die or present basal leaves blight, but they are not associated, were not associated to the viral symptoms. Also, the tobacco plants that we inoculate with the tomato leaves were also symptomless. And from the laboratory test, all the TMV immunohistochemistry were negative, so we couldn't detect the virus using this immunohistochemistry, so they were not present in the new tomato plants. And also the PCR performed by the right show a negative results for the leaf and substance, except for one line, this line 2, some of these bags that plants show the symptoms of the virus, but the rest of the bags were negative for the TMV. So as conclusion of this trial, we can say that both methodologies, both solution, T1 and T2, was good, so it's not important if the infected material is fresh or these are semi-purified virus. The fact that include the infected roots and leaf, the infected plant material, help to increase the temperature, so it's a good point for the solarization process. And also, if there is an area where the temperature can reach more than above 40 degrees, it is possible to use the solarization as a disinfection method. But this temperature must be extended to at least the time of the trial, at least 60 days. So now I'm going to present the result from the steaming method that were conducted by Richard Group from LNW in Germany. So they perform the steaming trial, and for that we have the same structure. So we have a pre-steaming stage, and a PSA after the steaming process. So they also use the susceptible tomato virality monomaker, the semi-purified virus, TMV, and they also evaluate the disinfection method on cocoa peat growth bags. So when they inoculate and transplanted all the tomato plants, they removed them, collected all the bags, and placed them in the steaming machine. So they tried 3 different incubation times, 20 minutes, 40 minutes, and 5 hours, and they also distributed the bags in one layer, and also they piled them in order to see if the position of the bags are important in order to have a good steaming process. And they also conducted a post-steaming trial in order to see if the disinfection was okay in the bags that they were steamed in the machine. And they also check all the symptoms by visualizing them and also using the ELISA test. So the results that they get is that symptoms of the virus infection appear on plants that do not steam substrate 3 weeks after the transplantation. But in plants that grew in steaming substrates, they didn't show any symptoms until the end of the trial. So they say that maybe there are some particles active in the substrate, but the plants grew really healthy for a longer time. And also all these results was confirmed via ELISA test. And also the other thing that they observed during the steaming process is that this method affect the microbiome of the substrate. So after the process, they observe a heavy growth of fungi and bacteria, potentially harmful. So they highlight that we must be careful with the microbiome if we are using this kind of disinfection process. So at the conclusion of the trial, the steam at above 90 degrees is a feasible method to deactivate the tomovirus. They say that if the dry heat, the trial that they place all the bags like 5 in more than 1 layer, so the disinfection, the steaming process is dry instead of moist. So it is necessary to do a long exposure or higher temperatures. So that means that the moisture enhances the effect of the steam. And with 20 minutes is enough for the disinfection process. We also conclude that the heat had an impact on the microbiome on the medium, so they suggest to apply beneficial organisms in order to avoid the growth of potential harmful fungi and bacteria. So it is best to use the substrate immediately after steaming instead of keep them for a long period. And finally, the effect of the steaming needs to be tested for every substrate. So this has been conducted in cocoa-peat substrate, but to know more about the real effect of the process, we need to test in different substrates. So this is all. I want to thanks to my colleague from Tenova that conducted this sterilization method trial, and also people from LNW to Murcia and the group for sharing with me all the results. Thank you for your attention. And also VIRTIGATION project for support us.
Question for Blanca?
Would commercial tomato growers normally reuse bags of substrates?
I think so. Yeah. In Almeria, there are a lot of growers that use the soil instead of the substrate, but people that use substrate reuse them. Not for a long period, but can reuse the substrate.
Of tomato brown rugose fruit virus on glasshouse surfaces and so on.
Ready to go?
No.
You can see.
Hello, my first class is very nervous. I'm Mary from Sarah, and just want to tell you about soil protection of ToBRFV and grass substances and soil. So, for those who don't know, Sarah is the National Reference Laboratory for Wales, who carry out a variety of commercial research work alongside our diagnostic testing. So at the minute, ToBRFV is still a regulated quarantine pathogen in the UK. UK surveillance at the minute, we've got 4 sites that have outbreaks. To give you an idea of how it's changed over time, when I wrote the abstract for this session, we only had 3. But all 4 are currently under eradication. So how does the UK manage to keep control of its ToBRFV spread? So on the left, I've got an image of a UK glasshouse grower. It's very dispersed. We had a talk recently where they were saying, could you give the locations of where the outbreaks are in the UK? If we said that in Kent, you'll know exactly whoever it was. That's how spread out they are. In contrast, here we have a site in Almeria in Spain. Everything's very close together, compact. You can imagine the virus goes very quickly. So we know from Anna Skirteffer's work on survival that the virus can survive on skin and gloves for over 2 hours. We also know that it can survive on glass cell surfaces for up to at least 6 months. If you ask anyone on the team about this work, we'll say we only stop testing at 6 months. So we have a very persistent virus. In a diagnostic point of view, how do you avoid contamination? Well, we could do things like bench coat, double gloving, disposable clothes, disposable sleeves, UV, decontamination. But even then, you can still detect fragments of the virus when it's swapping. So you can imagine in a glass house situation where you can't control these things, how it might come up. So here we have the swab results of a outbreak site in the UK. Unsurprisingly, the virus is everywhere. What is a bit surprising though is that we've also detected on the roof structure where the plants aren't actually coming into contact with the roof. So how is it getting there? We know that TMV and coronavirus can present in fog, and we've also, from Natasha's talk earlier, ToBRFV can be transmitted in water. So the next question is it airborne? Is it environmental residues? So Amy Flaps in our team has been looking into the sources of these environmental residues. So very quick overview, but how do you separate a plant from any potential environmental contamination? You build a really big box. So what we've done is we've swabbed from points around the box 2 weeks after setup, and the results are quite sporadic. So whatever is driving these detections is quite random. In some cases we've got CQ27, in other cases 34. So next step is very much going to be more replicates and is it actually valuable. ToBRFV is very much a global problem, so it has a global solution. So the FRESCO project is looking into the survival of ToBRFV in soil. So there are 3 key work packages: detection, survival, and then eradication. We spoke earlier about ToBRFV in soils. This is aiming at looking at sort of increasing replicas, and in an ideal world, we would ship out like 140 litres of soil to different project partners, and they'll carry the same methods, but it's not very easy to ship out 140 litres of soil to 7 countries. So instead, we have a generic potting mix where we have mechanically inoculated the plants with ToBRFV, allowed it to go out, and then after 6 weeks, remove the plant materials. We have a stockpile of inoculum. This was the original diagram I sent out to project partners, so it's very intense. So we'll just set it up a bit. So each time frame, we sieve the soil to remove the roots, but also left onto the soil and set up tomato plants in this, which we then extracted by Kingfisher and tested using a real-time RT-PCR. Each time frame, we also took the sieved soil to be directly extracted by a Belgian power soil kit. The material was also directly extracted by Kingfisher and then was used in bioassays to determine any local lesions, which were then confirmed by ELISA. So what results do you get with a virus as persistent as this? Everything is positive, unsurprisingly. Now we bring it down to the CQ values. So in the interest of time, I'm just going to skip past these. So as I said, this was a global project with actually a lot of the partners were moving down with us. So we wanted to, it's quite a time-intensive method, so we wanted to increase the number of replicates. So we start with the, I've got data by here, thank you. Kirsty Antonio from Italy, Irina from Slovenia, and Serpa from Turkey. So we start with the direct soil results. We are still waiting for the power soil kits in Italy and Turkey, so we just have Slovenia and UK results. We are still detecting CQs of 18 at day 200 in the soil. How does this translate to roots? There's a slight decline over time, but we are still getting low lesions in that case, more than 30 at every time point. You'll notice that day 200 results are not on the graph. This is because there weren't actually any visible roots material left, but I'll come back to the end. We have here the results of the extracted tomato leaves from the on-sieve bait tests over time. So there's quite a general upwards trend as you move on. So these results from 4 weeks post bait test setup. For the 2 outliers at the top. I think they then became listed at a later time point. When you compare this to the skid rate test results, however, these are a lot more sporadic. So is it the root material that's driving the results? If it was, then why are we still getting CQ of 18 with the direct soil extraction P200? So the next steps are to consider how do you translate results to a biologically active soil. So we partnered with an organic grower in the UK and we'll graphic this study again. We also think about when we see decline of viable ToBRFV in the soil. What we're doing is we've got a peach sheet in a minute because we would have no root material left to actually carry out the bioassays. So ULIN, in the UK, is looking at alternatives to determine viability of ToBRFV. So we've taken the extract from day 200 and carried out a long range PCR and see if we can get a whole genome from this. And then finally, we've had talks today on solarization and steaming. 4% of the UK's tomato production is organic growers. So, you know, solarization in Spain, very possible. Solarization in the UK, not so possible. Similarly with steaming, organic growers are not willing to compromise like the biological activity of their soil. If you heat it up to X degrees, you then affect the microfauna. So that's something they're willing to do. So just want to thank everyone at Farah and the project partners, especially those who shared the results with me today. Thank you.
Thank you. One question because we are late. Yes, Anton.
Mary, you saw a very wide range of CQ values for the same sample. Were you thinking of spiking them with controls so that you can normalize the data? Because I'm wondering if it's due to the sample uneven mixed or if it's just because the different labs have different equipment, slightly different tests.
Yes, I think the initial inoculation material can be different between the different labs. I think Sevilla using the luciferase as internal control and spiking that. And yeah, there is quite a lot of variation. I think that might be more to do with the original inoculant material.
Thank you. Okay, the last talk, Daniela Weiss, that she will speak on tomato root system.
There you go.
Okay, thank you. First of all, I want to thank the organizers for allowing me to come here and present my work regarding tomato root system as a model for studying growth defense trade-off during virus infection. So let's dive in. Virus disease symptoms, which were described beautifully here, mainly described for the brown plant parts. Not surprising because those parts have economical and agricultural benefits. However, it seems like a huge part of the plant that I mentioned, and this is exactly the root system. So why study the root system? First of all, they have a key role in plant adaptation and response to various of internal and external signals. Their activity and specificity actually impact the whole plant physiology. Secondly, as described here, they're also the source for soil virus inoculum and pathway for ToBRFV transmission into the plant in infected soil. So from here, we ask, can the root system be a turning point in shaping viral disease progression and symptoms development? So what is known about ToBRFV and roots? ToBRFV invasion into the roots was described actually almost a century ago in Samuel work, which showed that after inoculum of tomato leaf with TMV, the virus first moved to the roots where it accumulates 4 days post-inoculation, and afterward move and spread to the above ground parts, especially young leaves. We can generally say that ToBRFV's systemic movement is largely determined by the photoassimilate zinc strings. This experiment actually was reproduced in our lab in a published work done by Michael Weissman, which showed that after infection of Nicotiana benthamina plants with the TMV expressing GFP, the virus accumulated again 4 days post-inoculation and afterwards spread to the whole plant. In this work, it also been shown that the ToBRFV accumulate in the roots and systemic leaves 7 days post-inoculation, which is a little bit later compared to TMV. And here we want to say, we want to gain a broader overview on what changes occur in tomato roots in response to ToBRFV invasion. To do so, we chose 2 ToBRFV species, tomato mosaic virus, TMV, and tomato brown roost rot virus, ToBRFV, which we know that overcome allergens, but also spread slower compared to TMV and TMV. And we want to examine the change occur in root morphology, transcriptome, and hormones. In our experiment, we use roots that have been harvested from plants that have been grown either on a hydroponic system or on soil and then really gently washed. We check change in development using wet and dry weights, but also using green result system, which includes scanning the roots and then analyze the image. And we got a really nice and variant morphological traits, such as length and surface area, tip number, and number of branches. So we scanned the roots of TMV and ToBRFV infected plants 7, 14, and 21 days post-inoculation, and we got this. In red, you see the control, in orange, the TMV, in green, ToBRFV. And you can see overall reduction in root development and complexity, which means branching, in both TMV and ToBRFV. But you also can see that in TMV, the effect is more, you can see the effect more earlier, but also in more severe way. So at this point, we want to see which pathway and gene actually involved in the change in root morphology. So we perform RNA sequencing on roots. We decided to do it like at a very early stage of invasion, 10 days post-inoculation. And we got this PCA analysis based on the results. And here you can see the replicates of the control in red and the replicates of the TMV in orange, very grouped and separate from each other, but the ToBRFV are very spread. At this point, to focus our study, we decided to continue only with the TMV to further investigation, for now anyway. So we performed bone enrichment analysis on the TMV infected roots, and we could roughly characterize the growth chunks into 4 groups. So first of all, stress and defense response include growth chunks for osmotic stressors and systemic viral resistance. Development includes cell cycle, cell wall, and root growth. Metabolism, including secondary metabolites of the phenol-phenylpropanol pathway. And hormones include auxin, ADA, salicylic acid, which I refer to as SA, jasmonic acid, sensory, sphingolactam, cervicine. All been changed. We were curious about the hormones. So we performed MSM analysis and quantified the hormone. On the same roots, we performed the RNA-seq, and the result as follows. You can see upregulation in SA and downregulation in free auxin and ABA, with no change in cytokinin and no detection of jasmonic acid. We found it quite interesting because SA and auxin have antagonistic relationship, that actually represent the growth defense bands in plants, in which high level of auxin induced plant susceptibility, but also growth and development. On the other hand, high level of salicylic acid increased pathogen resistance, but also reduce growth. So we ask, are the increase in salicylic acid level induced by the virus infection responsible for the reduction in root development? To try and answer that, we performed the next experiment in which we apply elevated concentration of assay to the leaf with or without combined treatment with ToBRFV. And then we check different traits of root morphology. And here you can see in the brown box plot, you can see the quite dramatic effect assay have on the root development. But what we found interesting is that when we combine the treatment, especially at the lowest concentration of assay, we get some additive effect. This result suggests that ToBRFV-induced salicylic acid may actually impact root development. Next, we want to see what happened to auxin. So we check auxin signaling in the roots. Also, we inoculate plants with expressing DR5, which is a marker for auxin, and we image the roots. And as you see here, you can see increase in root signaling in the root meristem. This result actually correlate with recent published paper show the same pattern in root meristem after applying assay treatment. And they also suggest that this is a result of upregulation of auxin biosynthesis and changing auxin transporter PIN1 and PIN2. So this result may suggest that the induction of assay by ToBRFV infection may impact auxin signaling in roots. So we went back to the RNA-seq results, and we want to see which gene actually changed in both hormonal pathway. And as you see here, I will just mention a few. For the assay, you see upregulation of biosynthesis gene, PAL1, and upregulation of pathogen-related gene, PR5 and PR1. However, in the auxin, you can see both decrease in the expression in auxin biosynthesis transporter and auxin signaling activators together with upregulation of auxin signaling repressors. So we validate this result in quantitative PCR, and we got upregulation in auxin signaling repressor ALF5, which is quite unique ALF, and several of auxin signaling activators, ALF5, ALF19A, and ALF19B, together with downregulation of PIN5, which is auxin transporter localized to the ER and regulate auxin homeostasis. We want to take it a little bit farther on. So we want to see whether those genes have any impact on root development in general in tomato. So we got several mutants from Professor Naomi Ori from the Hebrew University. And here you can see ALF5, ALF19A, and ALF19B, and we check their morphology and see overall reduction in root development and complexity, especially for the ALF5. And from here, I want to suggest some model, hypothetic one, which actually represents the complexity of the interaction between the virus and the hormones and the root development. And so, ToBRFV can, based on the literature and our results, can directly impact auxin signaling in biosynthesis. And this has actually increased plant susceptibility to the virus. For example, by allowing the virus loading into the phloem. ToBRFV can impact auxin through indirect pathway by suppressing RNA silencing pathway, which on one hand allow virus replication, but on the other hand actually interfere with regulation of different developmental processes. For example, through regulation of ARF gene. And we know that ToBRFV virus infection increase salicylic acid, which in turn has also impact on the RNA silencing, through for example, RDR, impact on oxygen transporters and oxygen signaling, for example, through activation and stabilization of OXIA. So to conclude, ToBRFV virus infiltrate the roots during the first week of infection. ToBRFV virus inhibits lateral roots formation and root branching. The effect of ToBRFV virus infection involves reprogramming of the root transcriptome and hormone profile, which we saw really nicely. And this reprogramming is characterized as a shift of oxy-mediated root growth and SA-mediated suppression of root development. And we currently try and characterize pathway and gene that involved in this growth defense trade-off. And I want to thank my lab members, especially my PI, Dr. Spiegelman, Professor Norbert from Hebrew University, and Dr. Ben Mansfeld from Washington University, which actually did the RNA-seq analysis.
Thank you. We are ready to schedule.